So I just turned it on. So maybe you can go to watch your.
Oh wow, it's right there.
Well, clear stage. How many followers? 200.
Hello, Charlie. Charlie's here. Hello.
Banana. Charlie's be on the screen.
I was on the stream.
On the screen. Yes.
But yeah, so yes. Okay, sorry. Sorry, Uncle Del.
Should I use sleep?
Yeah, maybe. Do you want to just maybe turn off the power saver and stuff like that? Like, so you won't turn it off by itself? You know, like after an hour or so?
Oh, I see.
Yeah, like actually, I think it's like battery saving or something, whatever. Yeah. Yeah. Why is it dark? I guess it's saving power. Again, I want to see if I'm on the...
What's up?
Okay.
What's the software called?
This one? OBS.
I can just do the recording and I'll probably just die.
Okay.
But you're recording also.
Yeah, it's really fine.
No, I'm sure it's fine.
No, it should do something. I think the sound is so we go back to you need to plug in your power.
It's because mine, this part is much shorter. Oh, I see. I will start it closer to. Well, I guess
Okay, hello, everyone. Welcome to the dissertation defense of Molly Gasperini, a PhD candidate in UW Genome Sciences. Molly's a 17th graduate student from our lab to defend her thesis, and the 5th dissertation this week, which is great, from the department. As is customary, I'll start with a few words in Molly's defense. That's a joke. Molly grew up 8 miles north of here in Shoreline, as she'll tell you, a mere stone's throw from the Costco parking lot. She became interested in science very early. I found this quote in the Seattle Times in the context of a fashion makeover article. Where 16-year-old Molly, and I think you'll all appreciate that the last sentence is just like something Molly would say now. So she hasn't changed much. Molly stayed local for her undergraduate studies here at the University of Washington. My understanding is that her first campus job was at the UW NASA office as an office secretary, but she sucked at it and was fired. So she also tried out acting. She thought I had good Google skills. How you like me now? Here, watch this. So this is from a full-length feature film reenactment of Jurassic Park that I found on YouTube, in which Molly acts. It's a long. I've taken the liberty of just clipping out about 30 seconds of choice clips, which are acting skills are showcased.
Amazing. Perhaps we could create a colony of ninja psychics. Not supposed to end this way.
So clearly she was more talented at that than as an office secretary, and we're glad she didn't go into acting. And instead chose a career in science. She, I think after that brief stint, she looked around for jobs in the lab and landed a position with Mary Claire King, where she spent several years working on schizophrenia genetics, receiving a number of awards for her research. While she was here, I'll just mention one, which was the Herschel Carroll Roman undergraduate scholarship, which is, of course, the undergraduate scholarship that is given by our own department. And I think this was taken in our lab, but I just love the quote below it. From an article in one of the UW papers. So after graduating from UW in 2012, Molly went to Lou Kunkel's lab at Harvard and Children's Hospital in Boston, where she worked for several years on Duchenne's muscular dystrophy in mice and zebrafish. She then applied to graduate school, and very fortunately for us, accepted an offer to come back here to Genome Sciences, where she joined in the fall of 2014. So Molly's reputation preceded her. I didn't actually meet her as an undergraduate, even though she was in our department, but she somehow already knew half of our lab from ASHG and other meetings where she got to know people. And in our first conversations, we arranged a rotation. But then I recall subsequent conversations where she wanted to rotate still, which I was excited about. But she also made it clear that she didn't want to join the lab because Andrew was likely going to join. And had, as everyone would agree, having a significant other in the same lab is a recipe for disaster. But at the end of her rotation, Molly decided that joining the lab was more important to her than her relationship with Andrew. And I remember a conversation where I was expressing, both being very excited to have her in the lab, but also a little nervous about having them both in the lab. That if it doesn't work out, it could be a disaster, that kind of thing. And she looked me firmly in the eyes, and she said, "Jay, don't worry, it's going to be okay." So again, I'm not going to comment here on the science, but I just want to highlight a few things about Molly that I've really appreciated. So first and foremost on the science front, and part of this echoes things I said last week, a few weeks ago about Andrew, but I really appreciated just the combination of energy, super careful and rigorous thinking that she brings to all of her work. Like all PhDs, this has been a journey of ups and downs and of moments, where it wasn't clear what to do next, and moments where it was crystal clear. Molly's traversed that, I think, by both a combination of thinking big, but also doing so in an extraordinarily rational manner, pivoting where it made sense to do so, pushing back against me when I was suggesting things that were not in line with reality or in retrospect made no sense. Most importantly, just by working really hard and also thinking really hard at the same time, right? She, is evident from that quote from when she was 16. She has lived and breathed science for a long time, and that's been evident in her entire time here. She's also brought, I think, an extreme degree, and I mean extreme in the most positive way, of care and rigor to her analysis, not only to her experimental design, but also the analysis and interpretation of data. Repeatedly kicking the tires, thinking of every possible alternative explanation for her observed data, and trying hard to systematically rule those out. And when you can't rule them out, just being honest and transparent about it. That level of care has been huge, and I think sets a standard for the lab, and honestly for an emerging field as well. And I'm proud not just of the work that she's done, but also all of that rigor and the transparency with which it's been presented in the papers that represent the work that you're going to hear about. The second thing I'd like to call out is just what a positive asset Molly has been to the lab. The lab is entropic, and a failure to do work results in a descent into chaos. When we're talking even about things like lab culture, and it's easy to focus on your own work and just worry about your own self, and Molly's never done that. She watches out for her lab mates and has helped the lab adjust itself in ways that are big and small, in ways that are visible, and also ways that are behind the scenes. She's never been intimidated by me, not afraid to call me out. Whether it's related to something that I've done that I wasn't taking, something that I'm not paying enough attention to, things I need to do better, and more generally just keeping her eye out for her colleagues. The last thing is that even as it's all about the science, it hasn't just been about the science. A running thread through Molly's time here has been her deep involvement with science communication, and I really mean this in a couple of different ways. So. The first is that she's taken the time to be involved deeply with a lot of things that, something that I think a lot of us pay lip service to but don't invest enough time in doing, which is improving science communication both with each other and with the public. This includes SciCon, which is a workshop that is organized by and for grad students where she's played roles both locally and nationally in the leadership and development of that organization, but also other contexts, including the Pacific Science Center, local science fairs, etc. She's apparently also a frequent communicator. These are the Slack stats for our labs, where she's racked up an astounding 55,000 message lead over Andrew, which is impressive. So her communication skills also come through in her presentations, where she's had the chance to give talks at an impressive set of venues, including a plenary talk at ASHG, Biology of Genomes, and other contexts, and where the resounding feedback I've gotten from multiple people is that her talk was not only spectacular, but also often the very best talk of the meeting. And so with those high expectations set, I'll pass it over to Molly. She'll have an hour till the end of the hour to present her seminar, and we'll have some questions from the audience, and then everyone's excused to go to S330, except the committee, and there will be a party up there. Things go well.
Wow, thank you, Jay. When you were talking about Googling people to find their history, I was really worried about that human velociraptor video. Glad you found it. Should we try a different? The computer screen is black, unfortunately.
I'm debating whether to throw your husband under the bus.
Oh, no. Okay, there we go. Okay, make sure my computer's awake. Okay. Slideshow. Okay. Okay, so thank you all for being here. It's the department's 4th PhD Defense this week, as Jay mentioned, or 5th Defense this week, so thank you all for sticking with it and joining me so early in the morning for a science work day. I wanted to start by spending about 10 minutes on a short layman's talk explaining why I think the work that we do here is so cool in this department. And so for starters, why is the human genome so cool? So fundamentally for me, it's because a human genome encodes a human life. So as an example, this is my nephew Charlie. He's about 2 years old, and he's keeping it together pretty well in the back. And like us all, Charlie came from a single cell. He came from a single fertilized egg. And like everybody else here, when we started with one cell, that tiny cell divided, and then it divided and divided and divided until there are millions and eventually trillions of cells that all work together now to build a really cute toddler. And so fundamentally, how did this one cell know how to do all this? And the answer to that is in the human genome. And so what exactly is the human genome? The best metaphor that I've heard of is like a cookbook. Individually, it's just a long string of letters, but together these letters make sense, and they encode instructions, and they encode recipes that we can then read to make many different types of food. But instead of meals, of course, the human genome encodes recipes to make an entire human heart, an ear, or even Charlie's whole brain. Remarkably, nearly every cell in Charlie's body holds a replica copy of that same cookbook. So it's not like the heart cell gets the specific heart recipes or the stomach gets the stomach recipes. Nearly every cell has the whole human genome within it. So how does each cell know how to read its specific recipes for eye or for brain, etc.? And that is essentially, that question, how does the cell know how to do all this, is essentially what I study, and I'll tell you about some today. But first, some more motivation for why I think it's so amazing to study the human genome. Charlie's genome contains his history. All the lives that have been lived before and come before him have contributed to his DNA. A little piece from every branch of all of our the giant trees that come and give us our own ancestry lie within almost every single cell in our bodies, inside Charlie's body. And the human genome also encodes some of Charlie's future, such as his predisposition for needing glasses, part of how tall he's going to be, or if he's predisposed to any illnesses. So fundamentally, I think if you want to study a big part of what it means to be human, you study the human genome. So to get cheesy with it, as genome scientists, we're not just biologists, we're in this department, we're historians, we're journalists, and we're also fortune tellers. So that's why I think the human genome is so cool, and that's why I wanted to pursue a PhD here. And so next I'm going to tell you a few, four of my favorite trivia bits about the human genome, and hopefully still in layman's language. Okay, so for starts, what exactly is the human genome? It's not just a paper cookbook, like I kind of led you to believe. It's a sequence, again, of molecular letters. And crucially, 99.99% of your own DNA is the same as a stranger's, which means we all have the same DNA as practically as every other human on Earth. We all have the same genes, and they're all largely organized in the same order and orientation and into chromosomes, as you have seen pictured here probably pretty often. And the human genome is that whole map of where all the genes should be and where all the other stuff should be, and that's why it's called the human genome, because we're all built from a copy of that same map. Okay, the next piece is that the human genome is both extremely old and extremely young. Humans diverged, if you want to count it this way, humans diverged from chimps 4 to 7 million years ago. That's when the first human-specific genome was around, you could say. But the map of it, or the sequence that the scientific community has put together, is only 18 years old, with the first draft being sequenced in 2001. And many of the members of this department have contributed to it. And so our sequence of the human genome was very young, and so there are still many mysteries that we're trying to figure out about it. And part of why there's so many mysteries is that the genome is vast. One copy has 3.2 billion letters. So even though we are so similar to a stranger, these numbers mean that in the tiny percentage that we do differ, there are still conservatively 4 to 5 million differences between you and that complete stranger. And so as we learned about in high school biology, most of our DNA's function is encoded in genes, and there are about 21,000 of those. So when we talk about the genome, we're talking about big numbers, thousands and millions and billions. But now for my last favorite piece of trivia, though we have 21,000 genes, 2% of our genome, only 2% encodes for those genes. So there's lots of in-between parts of genes that is called non-coding DNA sequence, and that makes up 98% of our DNA. And it was previously thought to be junk DNA, maybe not having much of a purpose, but by one statistic, 93% of disease-associated mutations fall in non-coding DNA. So what the heck is non-coding DNA doing? Why does a lot of disease heritability fall there? And it's clear that before we understand human genetic disease or the human genome, we need to understand this non-coding stuff. So spoiler alert, that's what I work on, non-coding DNA. And so in the last piece of the layman's talk, I'm going to tell you some about what this non-coding DNA is up to. Okay, so it's been known since the 80s that small non-coding regions can control when genes are turned on or off. And these non-coding regions are called enhancers. They're about 200 to 400 letters long, And in 2019, there are now thought to be at minimum about 1 million enhancers to be in the human genome. And we have many hypotheses for roughly where these are or where they might be, but these candidate enhancers are largely unvalidated and the target gene is largely unknown. And so why are enhancers specifically so important? Well, they act essentially as circuit boards to activate genes in specific combinations within the cell, so they can help create our highly specialized cell types. So in the brain enhancers help make sure that the brain genes are activated, and so on. In the heart genes are activated by heart enhancers, etc. And so, though I mentioned earlier that nearly every diverse cell in the body has the same copy of the human genome, enhancers are part of why part of that machinery that can control when and where certain recipes are read. So clearly they're very important, but they're also really hard to find. They don't really have a strict pattern or structure such that we can find them from sequence alone. They could fall many places in this vast non-coding human genome, and it's complicated by the fact that they are variably active across cell types. So how do we find them? Well, 2 years before I began my PhD, the CRISPR revolution began, and I was able to use the CRISPR system to make finding enhancers much more efficient. You've maybe heard, I know CRISPR is very exciting, Charlie, I'm sorry. Momo is my nickname, so bye-bye, Charlie. What exactly is CRISPR and how is it so revolutionary going to help us find our enhancers? Well, if you Google it, you get images like this. So maybe you think I've spent the past 5 years touching DNA, making stuff glow, and that would be super cool. Here's another artist rendering it a bit closer. We have this shadowy enzyme here. It's interacting with DNA, and many things are glowing. It's a bit closer. But what you in the audience need to know is that if I pare it down with my very simplified cartoons, these are the fundamental parts of CRISPR that make it so exciting. CRISPR makes it really easy to disrupt DNA in specific places. And the most important components of that are a pair of molecular scissors and a guide RNA that can disrupt DNA. And so the guide RNA just does what it sounds like. It guides the scissors to the place that it's going to cut, and that cutting disrupts DNA. And so then the last coolest thing about the CRISPR system is it's really easy to program the guide RNA with just a modern biological toolkit. We can do this disruption thousands and thousands of times in a single experiment, which is much faster than it used to be to run these experiments. And then, so for us, in my work specifically, the CRISPR system has made it really easy to efficiently disrupt enhancers, and that's great for us in searching the human genome, because if we disrupt an enhancer, we see, perhaps as a result, that its target gene is turned off, and then we can now have support that this enhancer likely targets this gene, and we've provided some functional validation for this candidate enhancer. So now we have an efficient way to look for them. So in summary, for my PhD work, I took the vast human genome, I searched for where there were enhancers in it, and I was enabled by this relatively new CRISPR tool to try to do this really, really efficiently. And hence my title, maybe, which hopefully makes a bit more sense to the layman in the audience, efficiently searching for enhancers and their target genes in the human genome. So that concludes the layman's portion of the talk, and so now for my performance art. And by that, I mean the rest of the talk is for molecular biologists. I'm not going to do some actual dance. I hope the layman watching in the audience can catch a few words, maybe just let it wash over you like modern art. Okay, so a quick reminder for molecular biologists. Part of why non-coding sequence is so hard to understand is that though maybe some of us are used to looking at DNA in linear space, in reality, of course, it's sitting in a highly complicated genomic and cellular context. And of course, DNA curves in 3D space in the nucleus, and if we even find non-coding variants in this region, we have to try and interpret them in this context. So there could be functional redundancy between enhancers. Enhancer A here may not affect the target gene at all, and it's just a red herring. But deconvolving this regulatory mess in the genome is going to be critical for every gene's regulation if we're to understand disease as well as human genomics. And so as I mentioned, CRISPR has come along as a new tool that's ideal for this. And it's ideal because, again, we can test candidate enhancers in their genomic context. And as I described, it's fortunately massively parallelizable, as guides can be easily ordered as a pool of synthesized oligos. So the little molecular biology, we can do these perturbations thousands at a time in an experiment. And this is clearly critical because, again, we have over 1 million candidate enhancers nominated by biochemical annotation. Okay, so for some last introduction, there are two main ways that, two main flavors of CRISPR that are commonly used to disrupt non-coding sequence. The first is the most common way that you've probably applied CRISPR yourself, where the Cas9 nuclease creates a double-stranded break, and a genetic scar is often created from that inaccurate repair, the cell's inaccurate repair of that cut. The other one is the use of Cas9 with an inactivated nuclease domain that's tethered to the crab repressor and essentially leaves the nucleic acid molecule alone, but tries to close up the chromatin. It adds histone modifications, recruits HDACs, and tries to induce heterochromatin with about 1 to 2 kb of the targeted locus. And this has previously been shown as an effective way to silence enhancers. And so these are the two ways I'll talk about using CRISPR today to disrupt regulatory elements. And for shorthand, I'm just going to be generically calling regulatory elements candidate enhancers. So I'll tell you about two tools that I've led development of in my PhD work. And they both use the CRISPR system, CRISPR-Cas9 system, to pair potential enhancers with their target genes. So first I'll focus on Scandel, work that was published about 2 years ago. And I actually started this project 4 years ago with the indelible Greg Finley during my rotation. Okay, so for a proof of concept for this study, we chose to try to find enhancers that control the Mendelian disease gene HPRT1, and this underlies Lesch-Nyhan syndrome. It's a rare neurogenetic and metabolic disorder that was first characterized in the 60s. Patients are severely impacted with devastating neurobehavioral and metabolic features. HPRT1 is a housekeeping gene, which is an important thing to keep in mind during this portion of the talk. And in a small percentage of patients, though they clearly have the disorder, they have no non-coding, or they have no coding mutation, meaning there could be some underlying non-coding mutations that could be contributing to HPRT1's disruption. And then also largely part of why we chose this gene to study is due to its role in the purine salvage pathway. Because of its role, loss of the HPRT enzyme leads to cellular resistance to the drug 6-thioguanine. So cells with functional HPRT die, and cells without it survive, which means we have very easy drug selection, very easy method to select for cells who have lost this gene's function. So for these main reasons, we wanted to better study and characterize non-coding sequence that could affect HPRT1. There have been previous efforts to do this. There was a plasmid-based deletion scan published in 1990, the year I was born, in mouse embryonic stem cells. And they found that the first part of intron 1 was critical to human HPRT function by creating 4 different deletions and phenotyping each one individually. They could then reason that only when the first part of intron 1 was missing was HPRT1 function impacted. But they only characterized 4 and on a plasmid, and so if we zoom out to about 100 kb on either side of HPRT1, and you put the previous paper's deletions in context here, this is clearly only a very small part of the surrounding cis-regulatory space. Instead, we wanted to phenotype thousands of highly overlapping deletions all around the HPRT1 locus and in their genomic context. So we did this. We performed just such a scan entitled over 4,000 deletions, such that on average 27 covered each base pair. And these, as you can see, these deletions were tightly staggered, so any signal could be hopefully dissected with high resolution. And we made these deletions in a high throughput manner by exploiting one of the editing outcomes that comes from double-stranded breaks. One such way we can make a long deletion is if 2 breaks are targeted near each other, at a certain rate you get dropout and deletion between the 2 cuts. And so in this way, we programmed our thousands of deletions and did this just by designing flanking guide RNA pairs. And so briefly for time, how do we create and phenotype these deletions? Well, we programmed and packaged our pairs of guides into lentivirus and then delivered them such that there was 1 deletion programmed per haploid cell. And so now we have a pool of cells programmed each to hold one of those unique deletions. And then to assess them, we simply just follow the now standard CRISPR screen format, and we sequence the prevalence of guide RNA pairs before versus after that HPRT drug selection. And then we just count up the occurrences of a guide RNA pair before versus after this selection using next-gen sequencing reads as our abacus. And so now for time, I'm also just going to cut to the chase and skip through our analysis and just show you the results. So here again is our entire HPRT1 locus, and here is the phenotyping data from the Scandel pooled screen. So we have critical sequence above the x-axis and non-critical sequence below it. And you can see the exons are coming through as positive controls, but in this test, we are largely not finding any distal non-coding candidate enhancers for HPRT1. And we confirmed this through directly sequencing the edits we were selecting for as well. We PacBio sequenced to validate that we were making the deletions we programmed, and we did confirm that only exonic sequence was coming through. Okay, so this is our overall biological finding for the HPRT1 locus. So there are few to no non-coding sites that match selective effects on the order of compromising an exon. Dodo is my partner Andrew's nickname. So we didn't find any candidate enhancers. In hindsight, this is reasonable because HPRT is constitutively expressed across many cell types as a housekeeping gene. Again, so one hypothesis circulating in the lit is that housekeeping genes don't actually require a lot of distal regulation, which our data support. And one possibility too is that we perform an extreme functional selection. So more subtle regulatory sequence perhaps is missed by our drug selection here. But in summary for this project, through validation, we confirmed that we successfully created a high throughput, massively parallel deletion scan, and we evaluated the Mendelian disease gene dysregulation in this cell type. We confirmed that validation is of course critical in these sorts of screens. But I think another huge take home from this work is that when I made the very first version of this plot at the end of my rotation, Greg and I were really disappointed, as you may expect. We didn't find any candidate enhancers. And then I spent 2 years showing that we really didn't find any candidate enhancers. And so we really owe a lot to Jay, I think, for helping me turn lemons into what I now think is a really cool story and project. So thanks, Jay. However, though we didn't find any enhancers, around the same time, other groups published very similar methods, and a whole slew came out about when we published. And again, these all rely on scanning Cas9 around a locus and phenotyping perturbations based on low versus high expression of just one gene. And other groups picked more dynamic genes than we did. They were luckier. They found many different enhancers. And so this method does work and can find distal enhancers. But our experience emphasizes, I think, how inefficient it is to perform one of these experiments for just one gene at a time. If we go one by one, testing all possible candidate enhancers or non-coding variants against a single gene at a time in one cell type, this is going to be impossibly slow since we have the whole scale of the human genome to chew through. So instead, ideally we'd be able to simultaneously test all candidate enhancers against all expressed transcripts in a cell to more efficiently get through all that possible enhancer gene pair search space. And so this was direct motivation for developing our next non-coding method. We originally termed it CRISPR-QTL mapping, and I'm going to tell you about that next. And this was published back in January. So Jay and I were actually directly inspired by an existing high-throughput method, which is massively parallel. It's a human genetics method. It's also massively parallel, and it tests many variants by all genes. So in these, naturally occurring variants from across human populations are leveraged, such that presence of a variant can be correlated with changes in any gene's expression based on bulk RNA-seq from tissues from these people. So then an association can be mapped just by correlating the presence of a variant with any gene's expression. And if a significant association is found, these sites are called expression quantitative trait loci. And one use is to hypothesize that somewhere in this variant's region is a candidate enhancer that's being disrupted by this variant that could control this target gene. And this method is extremely powerful because it allows you to resort the population and leverage one data set over and over again to test all variants you genotyped against all genes that you captured in RNA-seq. But as a method to screen enhancer gene pair specifically, these data are limited, as they suffer, the variants you've genotyped suffer from linkage disequilibrium, and we can only study naturally occurring genetic variation that falls in long haplotype blocks of many variants at once. So it's hard to parse out which variant is driving an effect, or if multiple are working together to do so, etc. So we wanted to take a step back and ask, what if we move this multiplex framework into the pooled enhancer screen perturbation paradigm that I just described to you? And so instead of human populations, we leverage a population of cells, and instead of mapping naturally occurring genetic variants, we induce large effect perturbations all around the genome using the CRISPR-Cas9 system. So here, a guide RNA library is just randomly distributed through the cell population. And then we can identify guide RNAs that correlate with reduced expression of a target gene in RNA-seq data from a cell to nominate that targeted enhancer and that repressed gene as a potentially functionally supported enhancer gene pair. And then we can also resort that population and leverage that data set over and over again to test all the enhancers we perturbed against all genes that we tested via RNA-seq. So this was the idea that Jay and I had been working on since I started grad school. And maybe in some horrifying insight, the first version for this was to do it with monoclonal cell lines and perform bulk RNA-seq on those cell lines to get the transcripts. So for context for anyone who's worked, I see some faces grimacing in the audience, because I was going to edit hundreds of sites around one monoclonal cell line and then do this 96 times. And that was just aim 1.1 in my general exam, 1.2 was 384 times, and those were just the pilot experiments. And so for anyone who has made one monoclonal cell line edited at one locus with a specific variant, this is clearly a very insurmountable task. And so I was slowly realizing this early in grad school and struggling to implement this with monoclonal lines, bulk RNA-seq, and I was in parallel watching Andrew Hill and Jose McFallon, and here they are pictured both with their babies. And they were performing really terrific work on adapting single-cell RNA-seq to the pooled CRISPR screen format. So as I learned about what Andrew and Jose were working on, I realized as an alternative, we should apply our multiplex enhancer gene pair screen framework to the single-cell RNA-seq format, because you never have to isolate and derive hundreds of monoclonal cell lines, and you can keep the cells in one pool and just use individual cells as individual data points. So what this would look like is, again, CRISPR-Cas9 perturbations are delivered here to cells in the pool, and the presence of a particular guide RNA can be correlated against all transcripts captured in the single-cell RNA-seq data. And so each data point here in this plot now is one individual cell. And to note, for all these experiments, we use the CRISPR-I epigenetic perturbation format that I mentioned at the beginning to avoid problems that come when we try to genome engineer multiple alleles in a polyploid cell line. So a few months after I was excited and switched to this, a paper with a very similar method, pretty much this, was published by Carrie Hahn's group in 2017. So of course I was really concerned, but then as Jay pointed out again, they tested only 71 sites with CRISPR-I with just one guide RNA per cell. And since single-cell transcriptomes are still really costly at this throughput, the throughput of this paper is still prohibitive. And so they have much less power than we do, because if you remember, we're multiplexing the guides per cell. And so if the guide RNA library is big enough, it'll always get a different, each cell will always get a different combination of guide RNAs, so we can treat each guide RNA in the population independently, and the effects of that one guide RNA independently. And again, the more we can multiplex guides, the more power is gained for each pooled screen experiment, and so we can get much more efficient use out of each single cell transcriptome. Okay, so we've been able to publish 2 relatively large screens with this method. There was a first pilot where we tested over 1,000 sites, and an at-scale experiment that I'll mostly tell you about today. And so in this, we screened over 5,000 sites in one experiment. We performed essentially over 78,000 gene enhancer association tests. We delivered 28 guide RNAs per cell and collected 207,000 cells to test them. And this experiment, if all goes well, it took longer than this, but if all goes well, it's designed to take about 6 weeks. So I'll take you through this experiment now. We picked, again, over 5,000 candidate enhancers in a K562 cell line. This is essentially based on open chromatin and other biochemical enhancer annotative marks. And then we designed 2 guide RNAs to each candidate enhancer to account for guide RNA quality. Then again, we made lentivirus of all these guides, and then infected the cells just at high multiplicity of infection, such that there were median 28 guide RNAs per cell. And then we collected 207,000 cells via the 10x Genomics single-cell RNA-seq platform. And so we genotyped the guide RNAs within each of these cells, the capturing and enriching for the guides nestled in a poly-A tag transcript here in the single-cell RNA-seq transcriptomes. And this guide capture work is derived from Andrew and Jose's previous efforts and project that we were inspired by. And so for context from this, we saw perturbation of each site we targeted in 900 cells out of that 207,000. And to note, to get this power and experiment with one guide RNA per cell would have required 5.8 million cells, which on its own would be one of the biggest single cell data sets. Okay, so now we have all these cells, we have their guide RNAs and their transcriptomes. So now how again do we test each perturbed candidate enhancer for its target gene? Well, we again separate out the population based on one site at a time, and then between these two in silico cell populations, test for differential expression of all possible target genes that we've captured in single cell RNA-seq. We did this using monocle. And specifically, we mainly studied cis relationships here, testing each perturbed candidate enhancer against all genes within 1 megabase. And so in particular, I really want to thank Andrew Hill here for providing me, just in general the know-how and computational power and skills for how to do all this, because if I've been left to my own devices to sort all this out and assign guides per cell and wrangle monocle and do this for over 200,000 cells, it would have taken me at least an extra year, if not more. And Andrew was critical to many stages of this project, but in particular to this one. So thank you, Andrew, and Stella, of course. So this is our analytical framework. So now how do we know if it's working? Well, first of all of our positive controls are coming through. And so in our library, we included guides that targeted the start of 381 genes, and we set an empirical FDR of 10% based on the non-targeting guide RNAs in our library that should have no effect on transcription. And so setting this threshold, 97% of these TSS targeting controls are coming through. So this is great, but now what about our actual enhancer gene pairs? We performed, again, these tests with the same empirical FDR of about 10%, or up 10%. And then in this experiment, we detected 664 significant enhancer gene pairs as passing threshold. And these are the hits that we called in our screen. And so to note, these enhancer gene pairs were made up of about 600 candidate enhancers that affected 479 genes, as some candidate enhancers affected more than one, and vice versa. So these are our hits that we worked with, and they're available if you'd like to check for your favorite gene or your favorite candidate enhancer for potential function in K562 cells. But now, since this is one of the largest functionally supported enhancer gene pair data sets, we can also, just besides one-off hits, we can also provide some of the first plots of an actual functionally supported distribution of distances between enhancers and their putative target genes. So now on this plot, just an example, if you have just one gene that's connected to a candidate enhancer about 250 kb away, it'll fall here at a 250 kb away point. So now I'm going to show you a null distribution. So this is all candidate enhancers that got paired with a target gene, but it's the distance between it and any gene within a megabase. So this is just the null. But now take a look at the actual set of high confidence enhancer gene pairs that we got from our data. You can see they're clustered near their target gene, but they're falling still a median 24 kb away. So if I were to zoom in, they're not scrunched up immediately close to the TSS, but they're definitely enriched for proximity in this whole potential megabase space. So additionally, we can get a sense if some enhancers always target their closest gene. Here I've plotted a histogram of the rank of a target gene's distance to its candidate enhancer. So what this means is that if, for everything that's marked in column 1, we had data supporting a candidate enhancer targeting its closest gene, whereas if it's in column 2, perhaps it skips over the closest gene or just targets the second closest gene, and so on, et cetera, for the third and fourth and all these other ranks. So you can see that 2/3 of them involve the target gene closest to the candidate enhancer, suggesting a strong role in proximity of enhancer to target genes, but still 1/3 of them involve skipping at least the closest gene. So overall, we've provided the one-off examples and then also functional evidence for genome-wide characteristics like this and more based on our large data set. Okay, so in summary for this project, we used our multiplexed enhancer gene pair screen to map perturb over 80,000 potential cis enhancer gene relationships across our 2 experiments. And for these, we associated hundreds of enhancers with the potential target genes, and we provided a functionally supported distribution of the distances between enhancers and their target genes. And I hope future iterations of this project will delve into why we're getting such a low hit rate from all the potential pairs we tested, as well as have further improvements in throughput, because again, we have so many potential enhancer gene pairs to test in the whole human genome across all cell types. So this is what I've been up to for the past 5 years. And overall, hopefully today, I've convinced you it's clear that we're approaching genome editing CRISPR methods that confusably assay massive numbers of non-coding perturbations both accurately and efficiently. And CRISPR screens and other high-throughput assays, as reviewed here by Leah, Jay, and me, are already enabling efficient characterization of the vast human genome. And so I need to do some professional thank yous to my partners on these projects, Greg Finley, who was fundamental to the Scandal project, particularly Andrew Hill, along with Jose McFallon, who was fundamental to many stages of the CRISPR QTL project. Jay, of course, I guess he helps them. And our funding, the entire Shendure lab, plus my committee for many hours of mentorship and advice about these projects and career and more. Okay, so now I want to take a step back and reflect on the evolution of what brought me here. So as Jay said, I grew up about 20 minutes drive from Fahey and Shoreline, and growing up, though I had really great hair, I actually had no intention of being a scientist. I really didn't like school up until probably 9th grade. No one in my family worked in science, so I had no exposure to this world or this career. But in 10th grade, I got even better hair. And I took a biology class from Ms. Tracy Stoops, who was the first science teacher just to completely blow my mind when we learned about human biology. And it clearly looks like she also inspired me to take up crafting, because I made a hand-sewn stuffed plush diorama of DNA that I'm sure I completed as a project for that class, not my spare time. So thanks to Ms. Stoops, I went to UW for undergrad, wanting to study biology. The hair's getting a little better. And you can see me a month or two, actually, before I started working in a research lab ever. And if you zoom in, you can see I'm studying DNA on that page right here for the Biology 200 class, if any of you who did undergrad here remember. Here I am as a senior at UW after spending 2 years in MC's lab in this department. I was featured in a Seattle Times article for undergraduate research, and I'm giving a really hard-hitting quote that says, "My goal is to help out in any way." So hopefully I've accomplished that. So here I am now, 2 years later, day one of PhD, starting with Andrew. Here I am last summer in grade 21 at this point for the kid who didn't want to go to high school or college. And I'm giving some of the slides you can see that I just gave you today in front of a really big audience. Okay, so I'm showing you this not to brag or tell you about my great hair, but because I think it's easy when you look at a resume like this or see a dissertation like this to think of it as a highlights reel. And easy to say, of course, she had success. But in reality, throughout all this, I sometimes didn't feel like it was going well at all. And if you're new in science, or if you have any sort of feeling or insecurities that you aren't supposed to be here, or that you're not good enough to be a science major at UW, or not smart enough to be in a research lab, or won't cut it in grad school, if you ever feel that way, science can be a really tough place because it's so competitive and intense. So maybe if you're listening now in the audience, and if you ever struggled with feelings like imposter syndrome, or feeling like you didn't know what you were doing, I want you to know that I felt that way a lot of the time going through all this. And this is in hindsight, what was a very successful run. So I still do sometimes feel that way, but if you do feel this way too, I want you to know you're not on your own, you should not be intimidated, and you do belong here. And working in science is amazing and so worth it. And if I can do it, so can you. And the main advice I have for you is if you're struggling with stuff like that, is to surround yourself with an amazing supportive team of people. And it's been a huge amount of effort into one person's science education, just my education, from hundreds if not thousands of people. And I've thanked them all here on this slide, and now I will go through them all. Just kidding, this would take forever. Jay was cutting word count for a paper and got to the acknowledgement section, and it was so long, he said this. So if this was the Oscars, the orchestra would have played you off long ago, but there's no orchestra now, so here we go. So, I'm just gonna give you an abridged version, but there are many people clearly who deserve a heartfelt thanks, so I really appreciate it. Okay, first to the entire Shendure lab, current and previous generations. I've learned, I tell people I received maybe half of my PhD, or maybe 60% of my PhD research education from Jay, but the rest I've learned from this really excellent, generous, brilliant group of people that you see all over the slide. Next, I want to thank the grad students of the Shendure lab. Again, I'm lucky number 17 to graduate, and if you take a look here, these are a really intimidating, smart, wildly successful group of people, and I was lucky enough to follow in their footsteps, and I learned from most of them directly, and also got a lot done in trying to live up to the bar you all set, so thank you. And I want to extend a ton of gratitude to the grad students who haven't graduated yet. I've definitely learned a ton from each of you guys, and I'm really thrilled by the work you're completing now. You can do it, and I can't wait to see you succeed. And so now I want to give a really heartfelt thank you to two of my biggest friends and supporters in PhD school. First, there's Charlie Lee. He's an incredibly powerful and critical supporter. Some really hard times in grad school, and we would have really helpful conversations like this. Charlie, my sequencing experiment failed, and he would respond with an existential soliloquy, which would actually be just the pep talk that I needed. And so thank you for this very technical advice. And so then I also want to thank Melissa Chasen, who's been another extremely powerful friend and confidant trying to navigate grad school together. And she would also often give me really heartfelt pep talks like this one. So thank you for inspiring me, Melissa. So next I want to thank three really key mentors, Darren, Beth, and Leah, for giving me countless hours and three really important lessons in science. Be rigorous in your kindness and your science. You should relax, girl, and you're going to slay. And I'll let you figure out who said what. It's maybe a little obvious. Next, I wanted to acknowledge and extend my gratitude to four really key scientific mentors who had the biggest impact on my day-to-day scientific skills. Whenever I sit down to perform an experiment or think about scientific concepts or design anything, I will always be thinking in my head, what would J.D. Kenny and Riza Grave do? They're all sitting on my shoulders thinking, sure, I do it right. So I've been really fortunate to work with all four of you, and I'm really grateful for your generosity in teaching me the fundamentals. Okay, of course I need to thank my family. When people ask me what my family is like, I tell them it's like having your own personal Fab Five life coaches from the Netflix show Queer Eye, if you ever watch it. So if you do watch it, you'll know what my life is like every time we get together. So I have 5 people who care about my problems, talk me through them, help me do my hair, pick out my outfits. My sister picked out my outfit today. And I'm so grateful to all of them for keeping me really balanced and grounded throughout my education. I'm also so grateful to the Hill family for adding to the brand and making it the Fabulous 9. Supporting Andrew and me is turning me into a total cat lady, so thank you. Okay, so next, here's what I prepared for Jay. After this nice intro, this is what I did for you. Okay, just kidding. You all heard a lot of really nice stuff about Jay, since I'm the 5th student from the lab to graduate this spring. But I want to be the one to tell you that Jay is an egomaniac who made us get a t-shirt with his face on it. Not 1, but 3 times. He made us wear it, he made Charlie wear it. He's crazy. Okay, no, I'm just kidding. Obviously, this is not Jay's fault. It's super weird, but it's just internet weird. This was my idea, and I need to legitimize this t-shirt. If you see any Shendure lab members wearing around the department, this is a real thing. It's the 3-wolf moon shirt. And after a happy hour one day with the lab, I was like, we should make the 3J moon shirt. And this amazing lab actually made it. And now we have these shirts that haunt our dreams. So thank you to such an amazing lab for making this. And thank you, Jay, for empowering me to just reach true heights of creativity. But in more serious territory, thank you for the incredibly generous environment that you've built, Jay, to do science. You've made us all more fun, intense, smart, creative, hardworking, smart, generous scientists. I'm really grateful for being invested in that way and for taking a chance on welcoming me into the lab. Am I forgetting anyone? I don't know. Oh, forgot the cat stuff. But actually, I need to thank my human husband, Andrew. People sometimes ask me, Molly, how do you do all this stuff, all the science, these other activities, and this travel and everything? He still seems so happy. Well, I have a superpower, and his name is Andrew, and without him, grad school or any adventure would make me pretty miserable. And I'm so grateful that we did this together. And I'll close with one last photo that I really love from our wedding. I think it summarizes the idea that behind this great woman, there's a great man. And with that, I will take any questions. Any questions? Josh?
Can you remind me how you got 28 guide RNAs into a cell?
Yeah, it's just we added a bunch of really high titer lentivirus into the cell.
28 different lenties in a single cell.
Exactly, 20 different integrants inside a single cell. And And we did some follow-up work to make sure the cells were okay. Essentially just added more and more virus until there were cells that were dead, and then the cells that were happy, I just proceeded with those. Nick? Thank you.
I was wondering about the efficiency of cutting in particular in that last project that you talked about.
Okay.
And whether perhaps low efficiency cutting might account for a lot of the missing enhancer pairs. Whether you've done any enhancer gene pairs, whether you've done anything.
Checking, yeah. So in the last experiment, it was all CRISPR-I, which is just nuclease inactive Cas9, crab repressor, sort of closing off the enhancer. But actually, I think that mode of perturbation might be why we're potentially missing so many potential sites, because that's just one way of turning off an enhancer. It's not as clean, I think, as true sequence deletion. So potentially there's enhancers that are perturbable by CRISPR-I, or it's just innately missing something. Yeah, so good question. Sorry.
You might have said this, I might have missed it, but when in your enhancer screen, what kind of magnitude of RNA changes did you see in disrupting the enhancers? And was there like a difference in the ones like completely, if some completely abolished gene expression?
Yeah, great question.
Small.
So here are thanks for the plan. Yeah. These are our TSS controls, which was sort of kind of like the most extreme effect size from CRISPR-I in our screen. And then here are the pairs. You can see there are some that are as extreme as you're expecting, repressing up to maybe 80, 75%. But many of them are just repressing at 25, 10%. And it would be really nice analysis to see if there's something different about the ones we targeted here versus the ones that just had a smaller effect size. Great question. Okay, I think that's it. Thank you all for being here. Yeah.